So today we're going to talk about Chapter 3, which is the cellular level of organization. The first thing that we are going to talk about is cell size. Okay, human cells are really small. Taking a biology course, you should know that you have to look at them using a microscope. And in order to see the detail in the cells, we have to use an electron microscope in order to see those. So for human cells, most range from about 10 to 15 micrometers in size. This is very, very small. We have some larger cells, like egg cells, and these are about 100, sorry about that, 100 micrometers in size. Again, these are not visible to the naked eye. They are really, really small. On the other hand, we also have nerve cells. We'll learn a lot more about these later on in the semester. Nerve cells are really long, and some can be over a meter long. This is because nerve cells can extend from the central nervous system down into the peripheral nervous system. So they are considered the longest human cell because of this. And we will talk more about this as we get into the nervous system chapters with that, okay? But with these sizes, we do have some limitations on cell size, okay? And this has to do with cells and their growth and their ability to grow as well. Okay, so for instance, cells can grow, and when they grow, they increase in volume, and there comes a point when their volume becomes more than their surface area. So these cells grow, increase their volume, and it becomes more than their surface area. So their volume to surface area ratio will change as they grow. This is something important to keep in mind, because if you think about the cell, remember the cell is a spherical, mostly spherical. We have cuboidal. There are cuboidal cells as well, and columnar, but they are 3D shapes, and as that cell becomes bigger, then their surface area becomes less. Remember, one of the things we had mentioned before was talking about the intracellular fluid inside the cell versus the extracellular fluid outside of the cell. And cells require the extracellular fluid in order to gain their nutrients to survive. So if a cell cannot get enough in from the outside, they are not going to be able to survive. So as the cell grows, it's going to have limitations once its surface area decreases, okay? So the bigger a cell gets, the less its surface area is going to be, and it's not going to be able to bring adequate supplies into the cell. So it's very important to think about that surface area to volume ratio. If a cell becomes too large, it can also rupture. Think about an overfilled water balloon. When you fill that water balloon too much, all of a sudden that water balloon bursts, and so a cell could do the same thing. So there are limitations to cell growth. The other thing that limits cell growth is cells that touch it, okay? So when cells touch each other, they stop growing. Okay, this is a normal phenomenon. It is cellular inhibition. There are inhibitory signals that are sent when cells touch each other. There are a lot of tissues in our body. We'll talk about these later on in chapter 5, I believe, maybe 4. Anyway, when we talk about these cells when we talk about these tissues, you'll notice that some tissues are very close. Cells in a tissue are very close to one another, and some cells in a tissue are very far apart from one another. When cells touch each other, they stop growing. There are inhibitory signals that are sent that cause them to stop growing, okay? So inhibitory, let's see. Okay, and those inhibitory signals cause them to stop growing. Now, the one thing that we know is that there are cells that can continuously grow on top of one another, and that we see with cancer cells. So there is no inhibition in these cells. They are not acting normal. Some mutation happened, and the mutation causes these cells to just grow uncontrollably. And when they do so, let's see, uncontrollable growth. And when they grow uncontrollably, they grow next to each other, they grow on top of each other, and they just continue to grow. So there's no on-off signals like there are in the normal cell. Okay, so we're not seeing the same thing with cancer cells that works in our normal cells. So normally our cells will stop growing when they touch other cells, when their volume gets too big for their surface area, because they want to be able to bring in the proper amount of nutrients so that they can continue to grow and survive, okay? Now, obviously there are times when cell death occurs. The cell dies, and when the cell death occurs, this will open up spaces in some of the tissues, and when we open up those spaces, then we again change the surface area to volume ratio, allowing there to be more surface area by opening that up, and so the cells can start to take in nutrients from the outside, grow more, and then divide. So cell growth opens up areas, changes surface area to volume ratio, and allows cells to take in. One of the things we're going to talk about later on in this lecture is mitosis. You've heard about it before, how cells can grow and divide. In order for a cell to divide and make more of itself, it has to be able to take in the nutrients from the outside. So once it can take in enough nutrients, take in enough water to get enough cytoplasm for two cells, then it can divide through mitosis. A cell won't normally do this unless a space opens up for that cell to be able to take in those nutrients, get big enough, and then divide. So when cell death happens around these cells, that gives them enough area for this to occur, where they'll take in more nutrients, they'll get bigger, and then those cells can divide out. The exception to that are there are some stem cells in the body we'll talk about throughout the course that are continuously growing and dividing based on where they are in the different layers of tissues. But for the most part, we don't see a lot of cell division unless cells in that area die off. Okay, let's talk about the separately, okay? So the first part of the cell that I want to talk about is the plasma membrane. So let me put this up here. Okay, so we're going to talk about the major constituents of the cell, and we're going to start with the plasma membrane. Okay, the plasma membrane is also referred to as the cell membrane, so you've probably heard of it referred to both ways. Okay, and when we were talking in chapter 2 about chemistry and different organic molecules, one of the things that we talked about were fats, lipids, and specifically phospholipids. So we mentioned this term phospholipids, and phospholipids are part of that lipid group that we were talking about, and they are the major portion of the cell membrane, which we had mentioned at that point. Okay, so the phospholipids are going to make up the majority of the cell membrane, and then within that phospholipids, we also have a variety of different proteins. Let's put this over here. We have a variety of different proteins that we're going to see within the cell membrane as well. Okay. This is the phospholipids make up a bilayer. You always hear about the phospholipids. That just wanted to spell correct. Let's fix that. You always hear about the phospholipids as a phospholipid bilayer. So if we think about the cell itself, one of the things that we see with this is that this phospholipid bilayer looks something like this, where we have two set of phospholipids, okay? So each one of these is a phospholipid. The entire thing is the phospholipid that we had talked about over in the chemistry chapter. So phospholipid. And in that phospholipid, we have the head, and this portion down here, which is the tail. Okay, and the same goes true for the one that is underneath it. So we see this phospholipid bilayer where we have one complete layer on top and one complete layer on the bottom. And what we had talked about before was that the tails themselves are hydrophobic, which, if you remember, means afraid of water, water-fearing. And the heads like water, they're water-loving. And that is hydrophilic. Okay, so the tails are hydrophobic and the heads are hydrophilic. The tails face each other, and that is why we create this bilayer, because the tails are hydrophobic and they face each other. The heads are on the outside, and the heads will touch either the extracellular fluid or the intracellular fluid, just depending on, we'll just do one on either side. The intracellular fluid, depending on, right, where the cell is. So one will wrap around the inside of the cell, and then the other will be touching the outside of the cell. So the heads are going to interact with the fluid, and then the tails are going to interact with each other. And the way this is set up is really nice, because the fact that the tail has hydrophobic properties allows certain proteins to be integrated into that plasma membrane, okay? Let's take a look at a picture of this, of the cell. So we all remember seeing this cell picture taking biology, okay? And so we see what the cell looks like in the different parts of the cell. And so let's take a look at each part of the cell and talk about each one of those. Picture than my drawing. Here's the cell itself, and let's flip over to this picture here. So here's a nice picture where you can see that phospholipid bilayer highlighted here. You can see how the heads are all aligned. Again, this is a nice picture because you can see that three-dimensional feel where the heads are all aligned on top. The tails then face each other. Okay, so the heads are then going to either come into contact with extracellular fluid being on the outside of the cell, or on the inside of the cell you would have some intracellular fluid. So the heads are going to touch those fluid portions, the tails are going to be facing each other, and then what you notice coming through the plasma membrane is a variety of different proteins. So there's all sorts of types of proteins that are here coming through the membrane itself. let's talk about some of these proteins that we're going to be able to see through the brain. Let's change slide. Here we go. So here's one, the ion channel. This is an integral protein. So one of the things we could talk about first is the difference between an integral protein and a peripheral protein. So you'll see this term integral here. And what that term means, let me pop up a text block. Integral means that it passes through the membrane, through the lipid bilayer, or obviously that cell membrane. And then another term that you're going to come into contact with are peripheral proteins. So there's a difference between peripheral proteins and integral proteins. And the peripheral proteins, just like they sound, they're attached to the periphery. So you're going to see those attached either to the inner or the outer surface of the membrane. Okay, so rather than being through the membrane, peripheral proteins will be on either side. So the ion channel, coming back to the ion channel that's shown on this picture, the ion channel is an example of an integral protein, and as you can see, it extends through, let's see, it extends through that phospholipid bilayer. So you see it extending through, which is why they're calling it an integral protein. Now these proteins are really important. We talk about these a lot later on, and their purpose is to allow things through. Most of them only allow one type of ion. Remember, we talked about ions in the chemistry chapter. So things that have a positive or negative charge, an anion or a cation. And if you remember before, we talked about their ability to make ionic bonds, in which case one takes the electron and one gives away the electron, and because of that interchange of electrons, some things will have positive charges and some things will have negative charges. So for example, we could talk about things like sodium or chloride, okay? And sodium and chloride are ions, and you see that they have the positive and the negative charge. So we talked about ionic bonds and how these elements come together, they exchange electrons, and then they dissociate in water. So whenever they're in the presence of water, they dissociate, and we said that was an important concept because we couldn't have ionic bonds in the body because we are mainly made of water, but we do see a lot of ions, and they're involved in a lot of different things. So these ion channels are proteins, and they extend through the plasma membrane, and usually they only let one type of ion pass by. And what's going to happen is you're going to have, for example, you'll have a sodium ion channel or a chloride ion channel or a potassium ion channel. And when it opens up, it will allow these ions to pass through. Now, these channels are unique because they can open and they can close. So they do not just remain open. They have the ability to open and close and just allow that type of ion to pass through in there. And we will talk more about those. So that's an example of a protein that you're going to see in the membrane. Okay. Another type of protein that you're going to see in the plasma membrane is a carrier protein. Again, this is an integral type of protein, which you can see there. And the purpose of this is to transport substances across the membrane. In order for that to happen, what you're noticing here is this shape right here is the actual protein, and these circles here, all of them, are whatever's being carried through. So that specific substance has to bind to the carrier protein, and then the carrier protein allows that to be put into the cell. So this is actually a very specific type of binding and a very specific way to bring things into the cell. Not anything can bind to these. They're very specific as to what they're going to bind and allowed to come into. Receptors, we will talk a lot about receptors. This again is an integral protein. You see the receptor through here. All right. Receptors bind to ligands, and that's a very specific binding as well. There are many different types of receptors, and there are very specific receptors for specific ligands. And so when it recognizes its specific ligand, then something's going to happen. Sometimes this can activate a series of reactions that happen within the cell itself. Sometimes this allows different genes to be expressed, proteins to be made. It just really depends on the interaction that's happening. But we'll talk about a variety of different types of receptors. Just understand now that they're integral proteins and they do bind ligands. Enzymes. Enzymes are a protein. They are both integral and peripheral. Sometimes you will see them here like they're showing you where they pass through the entire membrane, being an integral protein, and sometimes you will just see them on either the inside or the outside as a peripheral protein. What they do is they catalyze reactions. You've heard about lock and key fit. They are specific, right? The enzyme binds to a substrate, and so that's a pretty key idea there, where the enzyme bind to a substrate. And once they do, it's going to catalyze a reaction. So it's going to increase, or let's say speed up, a reaction. Okay, so it's going to allow that reaction to happen faster. So we see this in a variety of different ways. We'll talk about many enzymes, but understand that they can be either integral through the entire membrane, or they can be on the outside or the inside, making it a peripheral. So they kind of fall into both categories. Another type of protein are these linker proteins. They can either be integral or peripheral. So they can either extend through the membrane like you see in these pictures here, or they can be on either side. Just like their name implies, the purpose of these proteins, they are filament type proteins, and they act to either attach to the inside of the cell and bind things together within the cell itself, or they can work to participate in linking two separate cells together. So there's a variety of different types of linkages with these types of proteins. Another type of protein that we'll see is something called a glycoprotein. These glycoproteins work as cell identity markers, and these cell identity markers distinguish your cells from anyone else's. Okay, so they are unique to yourself. An example of this is the MHC protein. We actually will talk about the MHC protein in AMP2 when we talk about immunology. But these identity markers identifies those cells as your own, and they can also identify each individual cell as what that individual cell is. Okay, the inside of the cell is made of cytoplasm. Cytoplasm, which is a water-based component with organelles and a cytoskeleton. So within the cytoplasm, we see fluid, and that fluid is the intracellular fluid that we've been talking about. And then within that intracellular fluid, we have organelles, which we're going to be talking about them individually, organelles, and we also have a cytoskeleton. Okay, the cytoskeleton is really important. You normally don't see the cytoskeleton on pictures of the cell, but what this is it's made of many different types of filaments, and they extend all throughout the inside of the cell, and these filaments help anchor the cell together so that the cell maintains its shape. these filaments will link to those linker proteins and help that cell maintain its shape. And then all the organelles that we talk about in the cell are actually attached to the cytoskeleton. So think of the cytoskeleton just like its name sounds. Like our body has a skeleton that's made of bones. The cell has this cytoskeleton that we cannot see, unless we use a very fine microscope, the electron microscope, but we cannot normally see it, but it does hold everything together in that cell. So when we talk about the organelles later on, we're not talking about these random floating organelles all over the cell. They are stuck where they are because of the cytoskeleton. So the cytoskeleton is holding those organelles together and also providing shape and stability for the cell itself. So a very important part of the cell. Okay, let's put that up there so we can talk about another concept, and this is where we can talk about membrane permeability. We've already talked about this, so I'm just going to give you a quick overview of the membrane permeability. Look in your notes for the other concepts that we have talked about. Let's put this here, and we can talk a little bit about this. Okay, so membrane permeability, one of the things that we've talked about with the membrane is that it's semi-permeable. So remember, not everything that wants to pass through the cell can pass through the cell. One of the things that I like to say is that the plasma membrane is like the bouncers at the club, okay? When you go to the club, the bouncers decide who can come in and who can't come in, and that's basically how the plasma membrane is acting. It's allowing certain substances to come through, in large part by those ion channels, by those receptors, by those carrier proteins. Those are highly managing what can come through. And then, of course, other things can come through the cell membrane itself, but it really depends on how big it is. So it is semi-permeable. Not everything that wants to come through that cell can come through that cell. So then we can talk about the idea of being able to transport things through the cell. Okay, and talking about membrane transport, what we can bring through, what we can't bring through, and how the cell can be able to do that. So it is selectively permeable. And when we talk about membrane transport, we can talk about going from the extracellular fluid into the intracellular fluid, right? So going from the outside of the cell to the inside of the cell, and then we want to get rid of things like wastes. So we can also talk about going from the intracellular fluid then to the extracellular fluid. Okay, so membrane transport could include both of these different things, and we can lump transport into passive and active. So passive transport, the idea of passive transport is that it requires no energy. Okay, and what that means is that it's moving with the concentration gradient, or you can even say that it's moving from a higher concentration to a lower concentration. Okay, this doesn't require any energy for things to move with the gradient. Okay, so moving with the concentration gradient or moving from a higher concentration to a lower concentration does not require any energy. When ion channels open, this is the way that it's going to move if we're talking about passive transport. Okay, so no energy required with passive transport. Some examples that we can talk about with this is osmosis, and this involves the movement of water. Right, so the movement of water from higher concentration to lower concentration, or we can talk about diffusion, okay, and diffusion is the movement of substances. Same thing, the substances are moving from higher concentration to lower concentration. Okay, so osmosis being the movement of water, diffusion being the movement of substances, again from higher concentration to lower concentration. So they are both a part of passive transport. They require no energy. And by energy, we are referring to ATP, okay? So keep that in mind when I refer to energy, that we're talking about ATP, so no energy required for that, okay? And then on the other hand, we have active transport, and it's referred to as active transport because active transport requires ATP, okay? It requires energy in order to work, and the reason it does so is because we're going to go the opposite way. With active transport, we are going to move against the concentration gradient. Or we can say, in this case, we are going to move from a lower concentration to a higher concentration. Okay, in order to push something against the concentration gradient or move it from a lower concentration to a higher concentration, we're going to have to use energy, okay? So think about the idea of diffusion. Goes hand in hand with those concepts that we've been talking about with equilibrium. Everything wants to reach equilibrium, and things are going to kind of move around until they do. If things go from a higher to a lower concentration, that's normal, okay? Moving from a higher concentration to a lower concentration is something that's normal. So here's an example of simple diffusion here, where there was dye at the bottom, you put the water up on top, and as you let that just sit, it's going to move from that higher concentration. It was much more highly concentration down here at the bottom, much more highly concentrated down at the bottom. And as we let it just sit with time, you can see how it starts to disperse up the cylinder until equilibrium is finally met in this last cylinder. Okay, so that's normal, that's natural. It's going from a higher concentration to a lower concentration, dispersing itself out there, and it's not requiring any energy in order for that to happen, okay? Another example of passive diffusion is osmosis. So we know that osmosis involves the movement of water from a higher concentration to a lower concentration. So what you'll see here is they put in the middle of this U shape, what they've put in the middle here is a selectively permeable membrane, okay? And so on one side, on the left arm, all we see are water molecules, and on the right arm, we see water plus solute molecules, which are these little pyramid-looking things, okay? Now, we know that the cell is selectively permeable, so in this case, it's not very easy for solutes to move back and forth, and so it's a lot easier for water to move back and forth. Since there's no solutes on the left arm is much more highly concentrated with water than the right arm. And because water can freely move by osmosis, the water is going to move from the left arm to the right arm, which is what you're seeing in the second picture. So it's moving over to the right side, going from the higher concentration of water to the lower concentration of water. So this is passive movement of water, and the reason it's doing this is to reach equilibrium, because if enough water goes into the right side, then we can equal that out, okay? So osmosis is the movement of water, passive transport with the concentration gradient. This last picture, don't worry too much about. By applying pressure, they're pushing the water back into the other side to equal out the volumes. Okay, we talked about this in relation to isotonic, hypotonic, hypertonic solutions, which is what your homework was about. So again, you should be professionals at this by now, but so that you get an idea here, remember again, isotonic is equilibrium, so the outside fluid, the extracellular fluid out here, is the same as the intracellular fluid inside the cell, so equilibrium has been reached and therefore there's no movement. A hypotonic solution will be less concentrated on the outside, more concentrated on the inside of the cell. Because of this, there's going to be more water outside of the cell, and so water will move into the cell, causing that cell to swell or burst. In a hypertonic solution, the concentration of the fluid outside, and you can see how they've depicted it, it is darker blue than the hypotonic solution. So in a hypertonic solution, the concentration outside the cell is greater than inside of the cell. And so because of this, water will move out of the cell in this example, and that causes the cell to swell or shrink. Again, we've talked about this, and so this should hopefully be a review. An example of active transport is the sodium-potassium pump. We'll talk a little bit more about this in specifics later on, but the reason we use this pump is to help maintain sodium and potassium levels inside and outside of the cell. And in order for this to work, as you see here, ATP is required, okay? So in order to pump that sodium out of the cell, from the inside of the cell to the out of the cell, which is what's happening here, we need ATP. So because ATP is needed in order for this to happen, this is a form of active transport. All right, let's talk about something called vesicular transport. So vesicular transport is a process that moves large particles or molecules through the membrane in vesicles, and that is why it's called vesicular transport. These vesicles are made from the plasma membrane itself. So the cell does have to replenish its plasma membrane again and again as it uses these types of transport. So vesicular transport basically is transport in vesicles, and it can do so in a few different ways, okay? The overall way it can bring things in is referred to as endocytosis, okay? And endocytosis can be divided into several different groups. One of them is phagocytosis. The other one is pinocytosis. And then the final one that we will talk about, let me move this maybe. And the final one we will talk about is receptor-mediated endocytosis. Okay, so in phagocytosis, this is referred to as cell eating. So this is taking in large particles. In pinocytosis, this is cell drinking. So this is taking in fluid. And then receptor-mediated endocytosis is a specific type of endocytosis in where it involves a receptor and ligand binding, okay? And whenever we have receptor and ligand binding, this is a specific type of process that is happening here. So let's take a look at some of these pictures that I'm going to show you here. First, we'll take a look at the last one I just mentioned, receptor-mediated endocytosis. So in receptor-mediated endocytosis, we can see here that what we have up on here are receptors. So this little thing that they've drawn up here by itself is a receptor. And what you can notice is that down here in this area where it has the clarithin-coated pit, what all those are receptors, and they're binding to the specific particle that's there. So when enough particles bind to the receptor, it causes this part of the plasma membrane to invaginate, and that's what you see here, and it takes that part in. And what you'll notice is that when it comes into the cell, which we're right in here at this point, it comes into the cell in a vesicle. And so all of these fall under vesicular transport because the actual plasma membrane is wrapping around these molecules, pushing it into the cell itself, and then it's pinching off. So the actual vesicle is coming into the cell. Phagocytosis is similar except in this case, with phagocytosis, there is no receptor binding. The receptors that they're showing you here have already bound to it. Those receptors are not attached to the cell membrane, so it's a little bit different. So in this case, the cell is eating, and the cell itself it doesn't have to have receptors around it like they're showing you here. But what happens is the cell wraps itself around some particle. In this case, it could be a microbe, a piece of bacteria, it could be cellular debris, and the cell is going to wrap itself around that particle, and then it's going to bring it into the cell in a very similar fashion to what I just showed you, where it's going to invaginate, the cell is going to invaginate its plasma membrane, and then it's going to pinch off into the cell. So we'll have whatever it took in the middle, and then it's going to be surrounded by that piece of plasma membrane on the inside, okay? Phagocytosis and pinocytosis happen very similarly, but pinocytosis, instead of bringing an actual particle in, it's bringing in some of the extracellular fluid into the cell itself, okay? Some cells do this a lot more than others in there, okay? And then we have the final thing that we can have is the reverse of that with cytosis. And exocytosis is discharging the material from the cell. So we have exocytosis, okay, and this is discharging the material from the cell. This can be stuff that it took in and it did not use. This could also be wastes, the cell getting rid of waste, and this can also be things that the cell has made that it's going to put into the extracellular fluid, okay? So exocytosis is going from the intracellular fluid to the extracellular fluid. It's putting things out of the cell itself. All right, and this happens the opposite way, where the vesicle fuses with the plasma membrane and then with membrane and discharges the substances into the extracellular fluid. Okay, so they will be in exocytosis, they will be membrane bound, they will be in a membrane, then that vesicle is going to fuse with the plasma membrane, and then it's going to be expelled into the extracellular fluid. So the opposite basically of phagocytosis, pinocytosis, all right? All right, let's talk about the cell interior. Go in through and start talking about some of the organelles. One of the organelles that we can talk about are centrosomes, and centrosomes contain two centrioles, okay, and those centrioles are important in mitosis. Okay, in mitosis, this is where the mitotic spindle comes from, and eventually will be used to help split the DNA. So these are really important. You've probably have seen them before, where they create a 90-degree, so they'll end up at either pole. Okay, they're formed of different filaments that are in there, but they form a 90-degree, and we'll see them at either pole. So I'll show them to you when we talk a little bit more about mitosis. Okay, the next organelle that we can talk about the side of the cell is the endoplasmic reticulum, which is pictured here, okay? We see the endoplasmic reticulum in this area right here, and the endoplasmic reticulum is split into two different reticulums. So the endoplasmic reticulum, we have a rough endoplasmic reticulum, and we have a smooth endoplasmic reticulum. Okay, and you can see them in the picture here. This this portion here that is studded with ribosomes, up here kind of greenish portion, is the rough ER, and this one right here that does not have the ribosomes is the smooth ER. Okay, so this one has ribosomes, and this one no ribosomes. Let's move this over here so you can see it better. Okay. So the rough, what is the functions? It's important to know the functions of these. So the rough ER, the functions of the rough ER is that it is involved in protein synthesis. Okay, every time you see ribosomes, think protein synthesis. Okay, so protein synthesis is because of those ribosomes. We also see ribosomes free floating in the cytoplasm, and so we'll have protein synthesis there as well. But because we have these on the rough endoplasmic reticulum, we can have protein synthesis. We're also going to have storage of those proteins and transport of those proteins. Okay. On the other hand, we have the smooth endoplasmic reticulum, and the smooth endoplasmic reticulum is important for steroid production, lipid processing. Okay, it's also important for detoxifying certain drugs, and it makes all cell membranes. So when we talk about the phospholipid bilayer, the phospholipid bilayer is coming from the smooth endoplasmic reticulum. That smooth endoplasmic reticulum is producing the phospholipid bilayer and putting it out there into the membrane. It's also capable of detoxifying certain drugs, steroid production, etc. So if we talk about specialties and cells, one of the things that you're going to learn in this class is that there are different cells and they do different special things, and because of that they have more or less of these different types of organelles. So for example, with the rough ER and its protein synthesis, we would see a lot more rough ER in cells that had to do more protein synthesis. And an example of that would be neurons. Our neurons have to make a lot of protein. They release these different proteins into the extracellular fluid, and we'll talk about how those different proteins work as neurotransmitters. And so it's really important that neurons have a lot of rough endoplasmic reticulum. So we'll see a lot of rough endoplasmic reticulums in neurons. The smooth endoplasmic reticulum, a good example of a cell that has a lot of that would be the liver. Liver cells have a lot of smooth endoplasmic reticulum because that's where everything goes to be detoxified down there in the smooth ER. Okay, the Golgi complex. The purpose of the Golgi complex, and we see it here. The Golgi complex, first of all, I want to point out that the Golgi complex looks very, very similar to the smooth ER, okay? The difference, though, is that the Golgi complex looks like a flattened stack of pancakes is how they describe it. So notice how, let's see if I can kind of highlight this here. Okay, see how we have one, maybe that's not a good color. Let's do this one. Here we go. See how we kind of go in this area, and follow with me as I do this. See, here's one, here's another one, okay? Look at the picture in your book of the cell. And so like I'm showing you, they're very nice and flat and on top of one another. In the smooth ER, go back and look at the picture of the smooth ER. What you'll notice if you look at the smooth ER, it looks very much like a maze where they kind of interconnect with one another, and you don't see that in the Golgi apparatus. So I know they look very similar to one another, but if you look at that pancake flattening look, you're looking at the Golgi apparatus. If it looks more like a maze, you're looking at the smooth ER. So the Golgi apparatus, or the Golgi complex, like it says here, let's write the purpose of this. So the Golgi apparatus, or Golgi complex, either one is fine. They both mean the same thing, okay? Its purpose is to modify, sort, and package proteins. Okay, so after they go through the rough ER, smooth ER, then they can get sent down to the Golgi apparatus. And what it's going to do, it's one of the things that it can do is something called post-translational modification. Okay, a big fancy word for after translation. If you remember what translation is just the making of the protein. So after translation, some of our proteins get what are called post-translational modifications. So what that means is something is extra is put on that protein to make it work, usually a sugar, to make it work the way that it needs to work. So if a protein needs that, it has to happen in the Golgi apparatus. The Golgi apparatus is also responsible for packaging up these proteins to be shipped out of the cell or to be put where they need to be placed in the cell. So I kind of think of the Golgi apparatus as like UPS, FedEx, where it's packaging these things together and then placing them where they need to be. As you notice, when they come out of the Golgi apparatus, they come out in these vesicles, okay? See them over here. And those vesicles, if they're going to leave the cell, can then fuse with the plasma membrane by exocytosis, right? Exiting, so exocytosis, so they can fuse with the plasma membrane and then they can be released out of the cell. Again, we would see this in the neuron as well, where a lot of the neurotransmitters that are going to leave get put in secretory vesicles so that they can fuse with the plasma membrane for exocytosis. Okay, this picture kind of shows you how this is happening. We start over here in the rough ER, that's the green that you're seeing right here. The rough ER makes the protein, sends it over to the Golgi apparatus, which is here in orange. The Golgi apparatus can then just do any post-translational modifications, send it through, and then put it in these different vesicles. Okay, so some of them will stay in the cell itself, and then some will be transported out of the cell, and that's the different picture that you're seeing there. Okay, the next thing that we'll talk about are lysosomes. You can see the lysosome, the picture of the lysosome right here, and the lysosome is a package of enzymes, and they are actually digestive enzymes. Okay, so what happens is if something comes into the cell that needs to be gotten rid of, it can fuse with the lysosome. The lysosome can digest it up and then get rid of it. We see a lot of lysosomes in cells that are called macrophages. Okay, I just mentioned the word phagocytosis. Okay, macrophages are phagocytes or phagocytic cells, and what their purpose is to go around eating things. So they can eat things like bacteria, they can eat things like cellular debris, and as they take these in, they actually fuse that with the lysosome in order to break it up further. So there are a lot of lysosomes in these specific types of cells, and when we get to the immunology chapter in ANP2, we'll be talking a lot more about these and how they work in these specific types of cells. Okay, these lysosomes are actually produced by the Golgi apparatus. Another type of vesicle that's inside of the cell besides the lysosomes is something called peroxisome. Peroxisomes look like lysosomes, but they have different enzymes inside of them. So different enzymes than lysosomes, okay? And they are not produced by the Golgi apparatus. Inside of them, they have enzymes in order to break down excess peroxide. So any excess peroxide, hydrogen peroxide, they can break down. Now, I don't know if this is going to allow me to write this correctly. Oh, yes, let me see. So hydrogen peroxide is H2O, and there we go. So H2O2. So in cellular reactions, a lot of hydrogen peroxide gets made just as a byproduct of these different reactions. And we don't want those in the cell, because if we get a buildup of hydrogen peroxide in the cell, then that cell is going to die, because that's going to be lethal to the cell. So as the cell makes this, we want to be able to break this down. So it actually breaks down hydrogen peroxide into, let's see, it breaks it down into water, okay, so things that are not harmful to the cell, and oxygen. Sorry, this thing is not totally working with me. Okay, and ignore that period that it wants to put there. All right, so it breaks it, so it takes that hydrogen peroxide H2O2 and breaks it down into water, H2O, ignore the period, and oxygen, O2. So it breaks it down into less harmful substances that are not going to kill that cell. So basically it can help to neutralize free radicals. Let's get this in normal writing. Neutralize free radicals, and these free radicals come from chemical reactions that are happening within the cell. It can detoxify alcohol and other drugs, and it can break down fatty acids. Okay, and this is all important for the cell. Break fatty. Okay, so we see a lot of these. These cells are we see peroxisomes in all cells. Okay, so this is found in all cells, just like everything else, but it's very abundant in kidney and liver cells. We'll see more of these peroxisomes in those specific types of cells. The mitochondria. I like to talk about the mitochondria as the mighty mitochondria because it is important in synthesizing ATP. Okay, so with the use of glucose and oxygen, all those things essential for cellular respiration, going to the mitochondria, then we can make ATP. ATP is necessary in order for our cells to survive. It is the energy source of those cells, and we will talk a lot more about this as we go on, okay? All right, the nucleus. The nucleus of the cell is really important because in the nucleus is where we are going to have DNA. The nucleus is actually the largest organelle. It is the largest organelle, and it is where we will find DNA, okay, or deoxyribonucleic acid that we've talked about already a little bit. We'll talk a little bit more about it. So the nucleus is important. What you'll notice if you look at the nucleus here, we have in the nucleus, we have these areas over here. Let's highlight them. These things right here called nuclear pores. You see a bunch of them all throughout the outside of the nucleus. These nuclear pores are important because later on this is where mRNA is going to be able to pass through these nuclear pores. Other things can also pass through into that area so that we can have gene expression. All right, so the nucleus itself is surrounded by a nuclear envelope protecting that DNA, so the DNA in our cells are not just floating around wherever it wants to, okay? DNA is important because it contains our genes, those hereditary units that are within our body that codes everything for who we are, what we look like, etc. Different proteins that the cell needs in order to function. So if we look into a little bit about how DNA is packaged, what we'll notice here is that DNA is a double helix. We've talked about that concept before. It is a double helix. And then it wraps around these things called histones. Histones are proteins. And what happens is if DNA wraps around these, it can really tighten and condense itself. And that's where we get these chromosomes. So when DNA is tightly coiled and condensed, then it is referred to as a chromosome. When it is not as tightly coiled and condensed, we refer to it as chromatin. So you see those two words there, chromatin and chromosome. So chromosomes are tightly coiled and condensed. They look like X's. Chromatin, it's going to be much more loosely coiled. In a normal cell, the DNA is in the form of chromatin, where it is lightly coiled, loosely coiled. When a cell is getting ready to undergo mitosis and divide, that's when you see it taking that more tightly coiled chromosome X looking shape. Okay, so we do see both types in the cell depending on what that cell is going through. All right, let's talk about the difference between transcription and translation, because we're talking about the DNA in the cell, and I've mentioned several things to you before when we were talking about DNA and RNA, but let's refresh our memories on what transcription and translation is. You've went over this in biology, so I'm going to give you a brief overview. If you don't understand what I'm talking about, then please go back in your book, read over these sections in your book, and make sure that you understand these things. You will be tested on them. It's important that you remember these, that you understand them, okay? So the difference between transcription and translation. So transcription is the process of going from DNA to RNA, and specifically mRNA. And then translation is going from that mRNA to protein. Okay, so if we're going from DNA to mRNA, then this is going to be happening in the nucleus, and translation is then going to happen in the cytoplasm, because translation has to happen on a ribosome. If we're going to get protein, it has to be on a ribosome in order for that to happen. So then we have the difference between transcription and translation in there. All right, so let's look at some pictures. Here we have transcription happening within the nucleus. You see transcription is right here, step 1. We have transcription, so we're going from DNA to RNA. That's happening within the nucleus itself. The mRNA leaves through the nuclear pore, and then on a ribosome we have step 2, which is translation, where the RNA is going to become a protein. Okay, I know that students get this confused all the time. The best way I can describe this is transcription is going, when you transcribe something, you're rewriting something, okay? DNA, remember, we talked about this before. DNA is made of what? DNA is made of nucleotides. Okay, remember that? DNA is made of nucleotides. RNA is made of what? Nucleotides. Okay, with the exception, remember that thymine is not an RNA, it's uracil, so no T, we have U in RNA. If you don't remember that, reread your notes, please. And then proteins, what are proteins made of? Proteins are made of amino acids, okay? So here we go. So the best way to think about this is when you're doing transcription, you're rewriting something, right? And so when you're going from DNA to RNA, it's basically the same thing. You're rewriting the nucleotide to a nucleotide, but you're changing it ever so slightly, taking out the thymines and adding uracil. So that's transcription, going from DNA to RNA. When you're going from RNA to protein in translation, you are translating it. you are going from a nucleotide to an amino acid. So it's like translating a different language, okay? We have to turn those nucleotides into amino acids, and we do that through the process of translation. So if you can't remember it, hopefully that helps you remember that translation is RNA to protein, transcription is DNA to RNA. So a quick overview of transcription, because again, you should have seen this in bio. So we go into the nucleus. On that DNA, there is a promoter, and in order to synthesize that, we need RNA polymerase, and that is what they are showing you here. Let's a little bit bigger in this next picture. So again, we need RNA polymerase. So in order to go from DNA to RNA, you need RNA polymerase. Make sure you know that. You need RNA polymerase to go from DNA to RNA. And so we can open up that DNA, and hopefully you remember that in order to open up that DNA, we need DNA helicase. All right, an enzyme that opens up the DNA in there, and that's how we can get in there. Remember, the base pairs of DNA are held together by hydrogen bonds. I mentioned that the other day in class. And so we can open that up and we can get that mRNA. So we're going to synthesize mRNA from the DNA with RNA polymerase. DNA helicase opens that up first in order for that to happen. And then eventually we're going to get that mRNA out of it. Okay, the mRNA is going to go and attach to a ribosome. Remember, a ribosome is actually rRNA. Okay, so we actually have 3 types of RNA involved in translation. So now instead of transcription, we're at translation. I know I'm going through this fast. I do expect you to understand these things. If you don't, please read your book, okay? You went through these in biology. So in order for translation to happen, we need the mRNA. That's right here in the red ribbon. We have the mRNA. We have the rRNA, which is the ribosome broken up into 2 subunits, a large and a small. And then you also have the tRNA. Okay, so we have tRNA that's going to be involved in this process as well. So all 3 of them. So the mRNA comes and attaches to the ribosome, which is the rRNA. And then we have the tRNA that is going to come here. Now what the tRNA has is the tRNA has an anticodon right here. So our mRNA is read in 3s. So it's read by the 3s, and those 3s are called a codon. And then tRNA has what is called an anticodon, and the anticodon matches up to the codon on the mRNA. And based on that matching, it puts a specific amino acid. It attaches a specific amino acid. And so it will move in, attach its specific amino acid that is supposed to be there, same amino acid every time, and then it will move over. The next one will come in, and the amino acids will be joined together with peptide bonds mentioned before. So peptide bonds are going to attach these 2 amino acids together. Okay, when we want to attach things together by dehydration synthesis, we remove water. This is going to continue to happen again and again and again. And what you'll notice is that as every tRNA comes in and they switch positions, we're going to have this growing chain. So it starts out small, it gets bigger, 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 okay? And so we eventually get this very long chain of amino acids, and we talked about the primary shape of a protein versus the secondary shape of a protein. Okay, that's going to come along in there. So eventually we're going to get the entire protein out of that. And the protein is going to fold according to how that protein needs to fold. let's talk briefly about the cell cycle. So you've learned about the cell cycle in biology. It's important that you remember those different things that you talked about in the cell cycle. Now, not all cells are going to be actively going through the cell cycle. Some cells are in this phase over here, the GO phase. That phase is the phase in which the cells have left the cycle for a rest. Many cells, like our muscle and nerve cells, are permanently in this phase. Some cells can exit into the GO phase and then later on go back into interphase, which is shown here in purple. And some cells stay in the GO phase forever. Like muscle cells and nerve cells will stay there forever. They won't end up in interphase, okay? So it's GO are cells that are have exited and they are not dividing. But cells that are getting ready to divide will go into the cell cycle and it will go through interphase. So interphase is all this purple that you see here. We have G1 phase being the first part of that phase. This is where the cell is going to start taking in things from the outside. It's going to start growing. It's going to be metabolically active. The organelles are going to duplicate so that eventually it can go into the S phase where it can then replicate its DNA. And then from the S phase, eventually it's going to go into the G2 phase or the second growth phase. And that's where the growth continues and it's getting everything ready that it needs in order to divide. So the last final touches, any enzymes, any organelles, etc. that it needs to divide will happen in the G2 phase. And then from there, it goes through mitosis. Okay, so this other phase is mitosis. So a cell that is in this cycle of interphase, basically the interphase cycle is where it's going to be growing, replicating organelles, replicating DNA, finishing that growth. And then the mitosis phase is where it's actually going to be splitting its DNA to divide the cells into two. Right, so when we talk about mitosis, we have to talk about DNA replication. Now, understand that DNA replication is different from transcription and translation. Transcription is taking out a specific part of DNA to get a specific mRNA to eventually go through translation and get a specific protein. So we're not touching the entirety of DNA, just a specific part. In DNA replication, which is what is shown here, we are replicating the DNA in its entirety, okay? So this is DNA replicated in entirety. So we are making a complete different copy of that DNA so that we have two exact copies. Now the cell doesn't want to do this unless it's going to divide because it takes a whole lot of resources in order for this to happen. Okay. Now remember, for transcription we needed RNA polymerase. For DNA replication we need DNA polymerase, okay? So to get DNA from DNA, we need DNA polymerase is the enzyme that we're going to use. So DNA helicase, just like before, will open up that DNA, and then DNA polymerase is going to come in so that we can copy that DNA. And what you'll notice is DNA is referred to as being semi-conservative. Semi-conservative. And what that term means is this. Conservative. What that means is this. If you look at the picture that you see here on the screen, you'll notice that the original DNA is up here in purple, all right? We open it up with DNA helicase, and then we get these two strands that are coming out in here, okay? When DNA is finally made, it's going to have one old strand and one new strand per each DNA, and so that's why it's semi-conservative. The term semi-conservative means that it keeps a parent strand and then also has a new strand with it when it replicates. Okay, so DNA replication, we're replicating the entirety of the DNA using DNA. so let's get into talking about mitosis. So remember, interphase that you see on the screen right here, interphase is the phase in which the cell is growing, the cell is taking in resources, it's getting ready to divide, it's duplicating its DNA in these areas, okay? And then we get to prophase. Remember that prophase is the area where the DNA is tightening and coiling and condensing. So what you'll notice as we get through to the late part of prophase, let's see, condenses and coils. As you get to the late part, you'll see here in this picture that the DNA looks very condensed and coiled in here, because that's basically what's happening in this phase. And the other thing that we see is the nuclear envelope. Remember, the nuclear envelope is the envelope that surrounds the DNA, is diminishing, okay? So it's disappearing. And getting less. And you can see that in here in this picture a little bit better in the artistic depiction, where we're getting the fragments of the nuclear envelope. So the DNA is condensing and coiling, and it's looking like chromosomes, which is why you can see it. It stains a lot better when they're condensed and coiled rather than when they're just chromatin, not. Okay. Coiled. All right. Then we have metaphase. Metaphase is probably one of the easier ones to remember, because all the chromosomes line up here at the metaphase plate. So they all line up right in the middle. Here are those centrioles that we talked about before, okay? So the centrosomes, remember I had mentioned that term centrosomes before. Those centrosomes are going to be composed of those centrioles, so they lie at 90 degrees to each other. In the interphase, they're going to replicate so that we have two, and you'll notice that they go to either pole of the cell, and from them they put out this mitotic spindle, and they attach to the different chromosomes that are in there, and the chromosomes lay nicely at the metaphase plate here. So underneath the microscope, as you see in this picture, it's easy to pick this one out because of the fact those chromosomes are all laying very in the middle. And then what happens in the anaphase stage is where the mitotic spindle from those centrioles are now pulling the chromosomes to either pole. So they've separated each duplicated pair. So each chromosome were duplicates. Each piece of the chromosome was a duplicate. So they're pulling them to either pole, okay? And that's what you're seeing in these pictures here. They're pulling them to either pole. So I say this is like the sister phase, because the sister chromatids, remember the sister chromatids are the two identical pairs, and they're kind of out in V's, so they're saying to their sister, no, don't leave me. And that's the anaphase phase, okay, where they're going to either side of the cell. And then finally we get telophase. And telophase is the opposite of prophase, where the nucleus is reforming. The nuclear envelope reforms, and the DNA is uncoiling. Okay, so it's going back to being chromatin at this point versus chromosomes, okay? It's becoming chromatin at this point versus chromosomes. Now, mitosis, mitosis is referred to as the division of the nucleus. And that's what we've seen here in these four different phases, okay? The easiest way to remember them is by thinking of the term PMAT. PMAT, okay? And PMAT stands for prophase, metaphase, anaphase, telophase. And at telophase, we still have a cell that has two nuclei. So if we don't have the final division, then we would have a binucleate cell, which means a cell with two nucleus nuclei, which is not what we want, okay? So mitosis ends only in the division of the nucleus. We have not completed dividing the entire cell itself. And so in order for that to happen, we need something called cytokinesis. And cytokinesis is the very last step. Okay, so here we are in cytokinesis. And cytokinesis is the division of the cytoplasm. So in order to get 2 cells at the end of all of this, we need cytokinesis to happen. If cytokinesis doesn't happen, then we won't have this picture. So the other thing to note about mitosis is that in mitosis, you start with a parent cell, and then from that parent cell, we're going to get 2 daughter cells. These are the daughter cells, and those daughter cells are identical to the parent cell. Okay, daughter cells are identical to the parent cell, meaning if your skin cell does this, you're going to get 2 skin cells that look just like those skin cells. So that's a really important concept with mitosis. In meiosis, and something else you should have learned, so I'm going to go through this briefly. Meiosis happens in gametes. Okay, so we see meiosis happening in gametes, not in body cells like mitosis, but in gametes or sex cells. Okay, so this is a little bit different because what you'll see happening here is you'll see that we have the same basic phases that we had before, right? Prophase, metaphase, anaphase, telophase. So we have the same basic things happening. Now, before we go through prophase, the very first prophase, notice how we've separated it into meiosis 1 and meiosis 2. Before we go through the very first prophase, there is DNA duplication, okay? There is DNA duplication, and so when we go through the first time, we do split them. So if you go through all the stages, you'll see how they split very similarly. One of the things that you're going to see here that you do not see in mitosis is crossing over. I'm going to circle this. This is important. Crossing over, okay? Crossing over happens between non-sister chromatids. So you see what's happening here. And what happens is they exchange information. So when we finish off and we start meiosis 2, you'll notice that the division that happened to start meiosis 2, it divides that first time after meiosis 1. That division that happens they're not identical anymore because of this crossing over. When they cross over, they exchange information, they flip-flop information so that when we start into meiosis 2, they already look different from each other. Now, when we go through meiosis 2, which we're going to go through prophase, metaphase, anaphase, and telophase, just like we did the first time, but what happens here is that when we start meiosis 2, there is no DNA replication, okay? So DNA replication happens in meiosis 1. You have DNA replication before prophase 1. You have it. But before meiosis 2, there is no DNA replication. So if we look at what happens here in meiosis 2, we're going to go through all the metaphase, we're going to pull them apart, and then we're going to split them apart. So at the end, because of the crossing over that happened in meiosis 1, you have all these nuclei. Look at each nuclei. They all look very different from one another, okay? The other thing that you hopefully note is that it has half the number of chromosomes than the parent cell, okay? So the parent cell has half the number. All right, so here we go, and we have half the number of chromosomes that are in here. So here's mitosis, prophase 2, and then we have half the number. Here's a better picture. I like this picture better because you can see these guys. So here we go. They look very different. Here's 1, here's 2. These guys went through cytokinesis as well. You see they're not stuck together anymore. They went through cytokinesis. And now because they went through cytokinesis, we have these resulting cells. Look how they look very different from one another, okay? Mitosis. Mitosis has a mother cell, a parent cell. It makes 2 identical daughter cells. Meiosis this one that we're talking about. Meiosis has one parent cell. It goes through 2 divisions, and at the end we have 4 final cells, and they look nothing like one another. Not only do they look nothing like one another, but their end product are haploid cells. Okay, so their end product is haploid. So let me write down the big keys. Mitosis, you have mother cell makes 2 daughter cells. 2 daughter cells, all identical, and they are diploid. Okay, a normal cell is diploid, 46 chromosomes. Meiosis, 2 divisions. Okay, meiosis 1. In meiosis 1, we have DNA replication. Meiosis 2, no DNA replication. Therefore, we have 4 end products. Okay, and every end product is haploid, half the number of cells. So not half the number of cells, half the number of chromosomes. So 23 chromosomes. Put this up here where you can read this. All right, take a look at that. That is the difference. This is because when we talk about sexual reproduction, when the egg and the sperm come together, they have divided by meiosis, and it's important that they have half the number of chromosomes, because then when they come together, they will form the correct number of chromosomes, being 46, okay? And then they will start to replicate at that point. If they came together with both of them having 46, then we would have a cell with the wrong number of chromosomes, and then that would be an issue. So this is important. Again, if you don't understand these concepts between mitosis and meiosis, please